Dear student, this is Kuldeep Singh GAUR from Department of Zoology, Government Meera Girls College, Udaipur. In the series of lectures involving introduction of cell or cytology, today I am going to speak on endomembrane system. Many membrane organelles which are situated in the cell, they function in a coordinated fashion, creating a system called as endomembrane system. Not all the organelles which are membrane bound present in the cell are involved in the endomembrane system. Those which get involved are endoplasmic reticulum, Golgi bodies, lysosomes, and vacuoles. Among the exclusions are mitochondria, chloroplast, and peroxisome. These three, though they are membrane bound, have been left out of endomembrane system because their function is not coordinatedly associated with the endoplasmic reticulum and function of endoplasmic reticulum, Golgi, and lysosome. First of all, have some glimpse of endoplasmic reticulum. Endoplasmic reticulum is a network which spans from nucleus to until the plasma membrane. It is one of the biggest organelles. It has tubular structure, and this endoplasmic reticulum divides the cell into two parts. One is luminal compartment, that is the part which is inside the endoplasmic reticulum, and another is extraluminar compartment, that which is present outside the endoplasmic reticulum, that is cytoplasm. This is the diagram of endoplasmic reticulum. You can cite these ribosomes present. They are attached with their bigger subunits, and this is the nucleus. Sometimes the endoplasmic reticulum is continuous with the outer membrane of the nuclear double membrane, and these are the nuclear pores. Ribosomes are attached on the outer surface of the endoplasmic reticulum. Those endoplasmic reticulum which associates ribosomes are termed as rough endoplasmic reticulums, but some they don't associate with ribosomes. They are called as smooth endoplasmic reticulum. The rough endoplasmic reticulum, which is laden with ribosomes, is predominant in the cells which are actively engaged in synthesis and secretions. Then the smooth endoplasmic reticulum, it is devoid of ribosomes, therefore doesn't get involved in the activity of protein synthesis. However, it is actively engaged in lipid synthesis, the steroid hormones, and in animals, and other steroid hormones are synthesized by smooth endoplasmic reticulum along with synthesis of lipids. The second prominent organelle of endomembrane system is Golgi bodies. It was discovered long ago in 1898 by Camillo Golgi, and it was a densely stained reticular network like structure near the nucleus. It has flattened sacs called cisternae arranged parallel to each other, and their number is varied. You can cite that these are the cisternaes, and they are parallelly arranged, and this is the forming phase, means the Golgi particles or elements, they get associated at this place, and concave face is the trans phase, also called as maturing phase, also called as maturing phase. Here you get the vesicle which are laced with, or which are laden with the secretory material, which has to be secreted inside or outside the cells. Both the phases, they lie opposite to each other. The function of Golgi is packaging of materials, and it's delivering to its target, whether the target may be intracellular or it may be outside the cell. This is the diagram of Golgi apparatus. These are the cisterns present, arranged parallelly. The packaged material arises in form of vesicle at the trans phase. This explains why they associate with endoplasmic reticulum, because the proteins which are formed in endoplasmic reticulum, they just get out in form of vesicle from the endoplasmic reticulum, and they get coalesced with the cisterns at the forming phase, and after processing they emerge at the transverse phase. The proteins synthesized by ribosomes on endoplasmic reticulum are modified in cisternae of Golgi apparatus. Thus, the Golgi is the site for formation of glycoproteins and glycolipids. Now, vacuoles. Vacuoles are also membrane-bound organelles. In fact, they are single membrane-bound. Their membrane is called a stonoplast. They are filled with water. They may be filled with some sap or some excretory products or other material which is not useful for the cell. They are very, very prominent in plants. They are absent in animals, barring few small vacuoles which we call as food vacuoles are present. Otherwise, they are the characteristic feature of plant cells only. And in many plant cells, they occupy more than 95 or up to 95% of the volume of the cell, and just throwing nucleus away from the center and positioning it to the exterior part of the endoplasm. In plant cells, there are numerous material which are transported against the concentration gradient into the vacuole. Hence, their concentration is significantly higher in vacuoles than in cytoplasm. The material, what has been talked here, the material which is inside the, which is present inside the vacuoles, its concentration is much higher than the concentration of it in the cytoplasm. That means it has been transported actively inside the vacuole. In animal cells, we see contractile vacuole in amoeba for excretion and osmoregulation, and food vacuoles in many cells which are actively engaged in engulfing food particles or parasites. Another prominent organelle apart from the endomembrane system is mitochondria. It is the seat and site of aerobic respiration. It requires staining to be viewed, mostly genus green B, and number of mitochondria per cell is always variable depending upon the physiological characteristics and activity level of the cell. The shape and sizes, they are also variable. They may be sausage shaped or they may be cylindrical. Their diameter is around 0.2 to 1 micrometer and their length is 1 to 4 micrometer. Mitochondria are double membrane bound. They have outer membrane and inner membrane. The presence of two membrane creates two aqueous compartments in mitochondria. Outer membrane forms a continuous limiting boundary of the mitochondrion, whereas inner membrane forms a compartment filled with dense homogeneous substance called as matrix. You can see that this is the outer membrane of mito. This is the inner membrane, and inner membrane is thrown into cristae. These are finger-like folds which are created to enhance the surface area of the organelle. Cristae are, as I've told you, is the infoldings of the inner membrane, and outer membrane is a continuous one. The function of cristae, this infolding increases the surface area. The two membranes have their own enzymes. Mito are the sites for aerobic respiration and they produces ATP. Therefore, mitochondria is also called as powerhouse of the cell. Matrix has a single circular DNA and a few RNA along with 70S kind of ribosome. Why 70 kind of a ribosome? Because I've told you in earlier lectures that mitochondria arose from endosymbiotic association of bacteria with eukaryotes, and the bacteria are prokaryotes, and you know they have 70S type of ribosomes. Now, how mitochondria divides in cell and apportioned into daughter cells? It is by mode of fission. The last organelle which we are going to study in this lecture is lysosome. It's a membrane bound vesicular structure and formed by packaging from the secretions of the Golgi apparatus and rich in hydrolytic enzymes, and the many enzymes present in lysosomes, they are collectively called as hydrolases. The hydrolases are active at acidic pH and mostly they are digestive enzymes like lipases which digest lipids, proteases which digest proteins, carbohydrates which digest carbohydrates, and nucleases digest nucleic acid. So students, this is all for this lecture, and in the next lecture we'll be studying cytoskeleton. Thank you very much.